The Vaccines and Related Biological Products Advisory Committee meeting, which took place yesterday, discussed the future of the vaccines. Dr. David Wiseman was there and he presented to the FDA Council about his concerns going forward with the shots. And so, yesterday, after the meeting, Daniel O'Connor had a chance to sit down with Dr. David Wiseman to discuss what had happened. And so, for today, we're going to air that conversation. From Trial Site News, I am Adrian, and our episode is starting right now.
Today, we have the pleasure again to have on the show Dr. David Wiseman. He contributes from time to time to Trial Site News. He brings a wealth of experience in the world of drug and device development, regulatory affairs, regulatory operations. And, today we're going to again follow up on the recent Food and Drug Administration Vaccine and Related Biological Product Advisory Committee meeting. And we had some ideas in Trial Site what they would talk about today, but Dr. Wiseman actually presented today. So we're going to allow him to introduce the meeting, what happened, and we'll get into some questions. Dr. Wiseman.
Okay, hi, thank you for having me. Nice to see you again. So I can explain what happened. So this FDA meeting was convened a while ago, and it's been a long time in the making. FDA have been talking about this for over a year, going back April or so of last year, when they realized that there was going to be a need to update the strains, the variant strains for the COVID-19 vaccines, and a lot of talk about exactly how that process should occur, what sort of data should be needed to make the decisions, et cetera, et cetera. That April meeting of last year turned into June meeting last year, where they made the decision to recommend the use of the BA.5 4/5 bivalent vaccines, which were then subsequently authorized in September of 2022. That was followed in January of this year with a further follow-up as to, well, what exactly should our process be to continue along this path. There have been parallel CDC meetings that were going on, but they knew there was going to be a meeting around about June in order to figure out that they would need vaccines for the fall period. So this meeting today was to sort of make the decision, as it were, or at least recommend to the manufacturers which strains, if any, should be used as updates to the COVID-19 vaccines. And that was the nature of the discussion today.
Understood. And we're really referring to the formulation, correct? It's the same platform, same mRNA platforms, but updated formulation based on the new.
I wouldn't use the word formulation, Daniel. I mean, it's supposedly the same process. That's a whole nother story. And it's supposedly the same formulation. That's another story. But in terms of the actual strain of SARS-CoV-2 that it's targeting, that's the decision as to whether it's going to be Omicron or a version of Omicron or a version.
So that's understood.
Yeah.
Understood. So on that topic, did the FDA decide on? Did the independent committee that advises the FDA decide on a strain?
That they recommended to the FDA, that the FDA recommend to the manufacturers that they go ahead and produce a monovalent XBB 1.5, preferably, which is a derivative of Omicron. They develop a vaccine targeted to the XBB 1.5 strain, substrain of Omicron. That was the recommendation.
Okay. And that's what we wrote about the other day. We said that it would be that strain. That's what the World Health Organization is recommending. That's what the European Medicines Agency and the European CDC is recommending.
Exactly.
So with that in mind, you were able to present today. And I saw a video clip of it. I thought it was brilliant. I thought you did an incredible job. It was it was broad, but it was also deep. Can you summarize why you think that that we need to rethink with these vaccines? That it's just not working anymore. What's
Well,
your basis for that?
well. The big question is, I mean, okay, aside from the issues of safety and are they gene therapies and all those sorts of things, which are hugely important issues, which we've covered before, and we can go into. The overall philosophy is this a good idea to be chasing variants? Okay, and this is certainly not my innovative idea whatsoever. I mean, this has been talked about, by members of the committee. Paul Offit at the last year's meeting said something like chasing after. No, he was quoted in Time magazine, something like chasing after variants is a losing strategy. Because this is not like the flu vaccine, and this came up time and time again that we make these decisions every year round about the early winter time, midwinter time, as to which strains of flu virus should be targeted for the following fall winter season, and
Right.
and they're trying to sort of dance between, well, we're going to use the same model, but it's not really flu. And that was a hugely important issue today with lots of committee members. But to answer your question directly, we're chasing variants. And even, I'll show you the slides. Dr. Marks wrote an article in one of the major journals a few months ago. Fauci wrote an article. And so this is not making any sense, because first of all, these types of viruses have never been really effectively controlled by vaccines of any kind. That's what Tony Fauci said. And
That's correct.
And Peter Marks said, and you can put the slide up. Peter Marks said something of the kind, that this is not a long-term solution to be making incrementally modified vaccines. We need to have a much more durable kind of long-term solution. So
long-term, yes.
But
I think
here's the evidence.
Dr. Wiseman, I think you can share the slide. I think we've enabled that you can actually share.
Let's just do that.
I think we should show the audience the slide that you're speaking. I see it.
So the overall right. So the overall philosophy here is okay. Adrian can grab that. So this is Dr. Fauci writing in the end of early 2023.
Yeah, he wrote that article, and we summarized it, acknowledging that they've never been able to really control these viruses.
And he said, and not only that, how can we expect vaccines, especially when they're systemically administered, as in the arm and so on? It's not even reasonable to expect the vaccines to be able to do this. So that's Anthony Fauci, and then as you know, the now retired head of the allergy and immunological diseases section of NIH, and then Dr. Marks, who is the head of the Center for Biologics, and writing in was his JAMA? Where was this? Anyway, he said continuing on the current path of variant-specific vaccine boosters is inadequate, and it will require more than simply making incremental modifications on the current generation of vaccines. So that's a general philosophical point, but very practically, they knew this practically back in January, and when they had the January meeting, I provided extensive comments to their briefing document, which have now been posted finally on the regulations.gov, showing that even less within 3 months of the bivalent vaccines being authorized in beginning of September 2022, already there were papers. This is one of them, but there were many other papers that came out since then. This one is called "Alarming Antibody Evasion Properties of Rising, BQ and XBB Variants." And look at the dates that I've captured from the paper. It was received November 21st, so they'd only been rolled out September the 1st, and already they're writing this manuscript and submitting it what is that? 10 to not quite 12 weeks later, and they're saying that the BQ, which has now sort of disappeared really, but the XBB subvariants present serious threats to current COVID vaccines. Okay, so this paper was omitted from the FDA's discussion document in January. There were similar sort of types of papers like that, and they knew this even in January. And so the really big problem is, well, okay, why didn't they act on it? If it was important then, and certainly people have been dying and getting sick and so on and so forth. Thankfully, not at the same levels as has happened in the past. But they could have pressed the button and said, okay, Pfizer, Moderna, Novak, we want you to make XBB variants now, okay? And instead it's taken all this long. So there's a balance that the FDA are trying to a tightrope that they're trying to walk, and the tightrope is, well, yes, we want to make sure we get the right variants, but we can't overburden people with vaccinating every 5 minutes with a new variant, because it's just simply not going to be sustainable. People aren't going
to let's talk about this topic, and I really appreciate these slides, and your position is understood, okay? And what I would articulate is one of they've known that this approach doesn't work well, and Trial Site News probably by the beginning of 2021 was arguing continuously that you cannot control this particular RNA pathogen with vaccines, with new vaccines. It will not work. You will not control it. Maybe you'll help reduce some of the severity, morbidity, and mortality, but you won't be able to control the pathogen. Now, underlying all of this is important to understand before I go into my question about flu shots, is there was a consensus, Dr. Wiseman, among the World Health Organization, U.S., other governments, that they wanted to get 70% vaccination rate around the world. And that was their probably arbitrary measure to try to achieve what we call herd immunity, okay? So from the very beginning, there was this underlying assumption that we are going to control this pathogen with vaccines, okay? But to do that, that means you need to create herd immunity. You need to be able to stop the transmission. And we learned very early on, by March, April at the latest, 2021, that you had breakthrough infections, and that in fact, the durability of these products were in question. Now, so we've known for a long time, to your point. A question I have for you, you made a very important statement, and I kind of want to challenge you on this, or at least ask you a question. Why isn't this like the flu vaccine? Every year we have a flu shot. Every year the CDC, FDA goes through this process to identify the variant of the year, influenza strains of the year. They develop a vaccine. They range in effectiveness anywhere from not very effective to somewhat effective. They're not hugely effective, but they can help. Why is this different, Dr. Wiseman?
Okay, well, it's different for many reasons, and many people have made this point. First of all, flu influenza is basically seasonal. It's been predictably seasonal. That's the first thing. There is a reasonably okay track record. It doesn't always work. That they can look at whatever strains are circulating in the Far East in the sort of January, February time point, and predict based on the patterns that are known of spread, of transmission, that those strains are likely to be the ones, or very close to be the ones, that are going to hit the United States, 8, 9, 10 months later. So there is a reasonable predictability. It's not the efficacy. Sometimes at best is 50, 60%. Sometimes it's 10, 15%. And if
Correct,
a completely new strain comes along, in the meantime, then all bets are off. But there is there's a seasonality, and there's a geographic predictability in terms of spread. Okay, the point has been made by many people, including people at the FDA, Jerry Weir and Peter Marks, and many people have said, we recognize that this is not flu for all those reasons. The other reason is that the mutation rate of the flu vaccines is much slower than the mutation rate for the SARS-CoV-2 virus, sorry, the flu viruses is much slower than the SARS-CoV-2 viruses. So there's not, you don't have the same length of time that you can, okay, this make the decision, look at the data, go tell the manufacturers, go make it, and they can do their manufacturing things with some kind of testing. You don't have the same time, luxury, if you will, in those cases. So there's a very big difference in terms of that practicality. But one of there's a very subtle point here that's sort of going on, and that I think is playing out between CDC and FDA. FDA, I think, do understand scientifically those differences, and they said, listen, we realize those differences. This is not the flu, but we're going to try and use at least the general framework of the model of how we use flu, and so on. And in even today's meeting, many of the committee members were sort of bristling against the use of the word periodic updates, and the word season and the word campaign, and it has to be done by the fall. And they very much wanted to decouple it from flu. A, because it's different, and B, because they didn't want sort of a negative association. If this fails, that fails, whatever. They wanted to sort of keep them separate for good reason. And there was quite, it got slightly heated, and Peter Marks wanted the wording of the voting question to remain as it was. In the end, he agreed to remove the word periodic. I think the word periodic in the phrase periodic update or something, which sort of made it more palatable. But in the CDC language, I suspect that their thought process is, no, we want to tell everyone it's just like the flu, because we want people to not to be vaccine hesitant, and we just want to normalize this, and as if it's the flu. This is just a normal thing. Go and get your flu shot. Go and get your COVID shot. Give it to children. Give it to everyone. Okay, and so the CDC are not making that distinction. I think because of the vaccine hesitancy issue, the FDA are trying to nuance the distinction and say there is a practical reason why we do need to have a seasonality in terms of the rollout, because most people are confined over the winter months. There's other respiratory diseases, so we want to reduce the burden of respiratory diseases that are going on in the winter, which is going to reduce the burden on hospitals, et cetera, et cetera. So it's a very fine distinction that people are trying to walk, and it got heated slightly. I mean, people really felt genuinely nuanced about, upset about this, that we're going to make the right nuance. And I think that was a very, very important discussion that took place today.
So from that discussion, and I appreciate your answers. For some reason, I remember reading somewhere that in some cases it's stated that the influenza viruses mutate faster than SARS, but no. Regardless, they're different. They're different. So with that in mind, and those distinctions and that mounting conflict, is one side winning over the other? Is it the flu, Dr. Wiseman, or is it something else?
Sorry, is what the flu? What
is mean? Well, the consensus, the emerging consensus among the FDA advisors. Is it to declare that this is really different than the
the advisors say this is we need to make the distinction, and even within the FDA they're saying that. Peter Marks is saying, listen, there's a practical we understand all these scientific differences, but practically it reduces, from a practical point of view, in terms of the manufacturing gear up and so on and so forth, and the rollout and all the logistics. Practically, it reduces something to like how the flu is managed, but not because it's the same as flu. The CDC sort of want to make it the same as flu, but so there was, I think Peter Marks was, he was digging his heels in, and he realized, he was getting a lot of pullback, and he said, okay, I'll take the word periodic out of the voting question.
So taking periodic out it signifies that they're moving away from the flu. Is
it going to really make a difference in the end? I mean, it's still going to. So what they trying to say is that, listen, are we just going to have one meeting a year? We're going to meet every June. We're going to have the same discussion. Okay, XBB, BQ, whatever it is, and make the decision, or we're going to do it more often.
Right.
And Jerry Weir, who's the sort of lead scientist, I suppose, at the FDA, said, listen, when we talked about this a year and a half ago, we said we would need to meet at least once a year, if not more. And so we would need to meet as appropriate. So I think they realize that, and I think they will carry through with that. But the opposite side of the coin is that, why? My point is that, they had an opportunity to act on this 6 months ago, 5 months ago, right? And implement a strain change potentially much earlier than today or whenever they're going to announce it, as they did a year ago when they were talking about BA1 versus BA5, where Pfizer could have deployed BA1, which was on its way out, but there was some cross-reactivity, and they could have deployed BA1, pretty much June, July last year. And instead, they waited till September to deploy BA5, the BA5 bivalent. And my feeling is that Peter Marks recognized they just couldn't keep, giving new vaccines every few months and trying to get over vaccine hesitancy. So I think there's a very practical, can we sell it to the people kind of issue? People are just not going to keep lining up for vaccines. So they had to sort of balance the two kinds of things, and I think that's what on Peter Marks' mind. And I have to say, listen, I disagree with Peter Marks on lots of things, but I can understand that consideration very much, and I think it's much more nuanced and appropriate to think that. I mean, it's a difficult decision to make. I'm not sort of, I'm critical of it in a certain way, but I understand it, as opposed to CDC, who seem to just want to say, oh, that just tell everyone it's like the flu, and just and shut the hell up.
But let us remember that the CDC, the way it used to work, is that the FDA and their scientists and their independent committees determine what is safe, what is effective, and they then make those recommendations to the CDC. What's happened is that the politicization of this whole process has created madness, okay, in my opinion. Now, a very important point I want to drill into for a minute, and it's technical, and we may bore a few people, but that it is what it is. Today, there's this concept of validation that, when you develop a vaccine and you make significant changes, that you need to go back and do clinical trials, that you need to really verify the different components and how they work together that are safe and effective, but also safe, very importantly, because we're seeing safety signals, and we're going to go into that a little bit more. We write about that all the time on Trial Site, okay? So what is your argument? It's important for the people to understand why these vaccine products should go through clinical trials, why they should not just be updated with no human clinical trial data.
Okay, so in terms of what's going to happen now, is that FDA in the next few days, I suppose, are going to make an announcement. They say, okay, we're going to instruct the manufacturers to go make XBB 1.5 whatever vaccines. Okay, and the question was asked by Dr. Offit, what sort of package, okay, are they going to need to present, in order to get approval or authorization? And so it was a sort of very cursory answer. Yeah, they'll have to present the package of safety and efficacy and blah 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 blah. Okay, now what actually happened today was that the only company out of the three, the J&J is out of it completely, but Pfizer, Moderna, and Novavax. So the only company that has actually done clinicals with XBB something is Moderna, and they've done small studies showing immunobridging, or antibody level cross-reactivity to these different variants, et cetera. So, but I would hope that by a couple of months' time, that all the companies would have done some level of clinical testing, but it's not going to be a 40,000 patient study. It's not going to be thousands of patients in a safety study. It's going to be maybe a few hundreds of patients, and right. So that's what it's going, and plus animal data. So the problem is, as we've discussed many times, the safety signals that have accumulated, with the original strains, the BA5, the bivalents, and so on, the safety signals are coming out of everywhere. And
yes, they are.
And FDA and CDC are just saying, well, there's no safety signal. We've got myocarditis. We see a few little things here, but then they say, well, it's really not. And ischemic stroke, it's really not a problem. And that was one of the couple of slides I presented, and you've seen those before. I presented at the National Academies meeting that was a couple of months ago, where they're supposed to be looking at safety signals. So I don't anticipate this new variant to be much different. There might be some subtleties, but again, it's the platform. These are gene therapy products. I mean, we are asking the body to produce in an uncontrolled, undefined way spike protein in all the cells of our body where the lipid nanoparticles distribute to. The question was asked a year ago, and I think I've got it here, and I'm sure we've talked about it before. I think it was exactly a year ago at the Burbank meeting that Dr. Paul and I said, what's making these? I think I deleted it from the slide deck. But he said, what's making where are these spike proteins? Where's the protein being made? How much is being made? Can you correlate it to how much micrograms dose of the vaccine you're using? And he got dismissed. And Pfizer says, well, that's just an academic question. It doesn't really matter. We've got the clinical data, and all you need to know, produces lots of protein. And we've talked about that no, this is absolutely a fundamental issue. If you can't tell us which cells and for how long they're making these spike proteins and where they go, okay, you shouldn't be asking anyone. This is a fundamental problem. And again, in the presentation that I'm sure you'll link to, we allude to that, and we've alluded to it times that they've missed out the genotoxicity studies and those kinds of things, and they've dismissed all of these things. The gene therapies, disingenuously, they're saying, well, it's not really a gene therapy because it doesn't integrate into the genome. Well, no, that's not what a definition of a gene therapy is. If you look at the FDA's definition, it says something like it could act by a number of ways, including by translation, okay, and it includes any nucleic acid, not just DNA. So these are gene therapies that they are excluded from the guidances because they are for infectious disease applications. But if it was for another application, other indication, inborn error of metabolism, cancer, whatever it would be regulated as a gene therapy.
Well, and on that note, we've done some research on that. Conducted some little study here and there, and wrote a few articles. And sure enough, Moderna itself classifies them as gene therapies as recently as 2020. So
right. So you're right. So in their 2020, one of their SEC filings.
Yes.
Something like, I forget the exact word, but something like these products will be regulated as gene therapy products or something along those lines. That was in 2020. I think they'd already started their clinical trials. This wasn't like, oh, we're gonna file with the FDA in a few months and then we'll see what happens. They must have already been talking to the FDA. They must have been told at some point, yes, they're going to be gene therapies, and then somewhere early to mid 2020. It all changed. So no, we're not going to call them gene therapies. We're going to call them vaccines. Yeah.
I think
so something must have changed
with the emergency. There was some change that happened that, where they created a much more narrow definition of what a gene therapy is.
No. The definition actually has existed for a long time, and going back years and years and years. Going back many years. It defines gene therapy. Include that if you just read the definition it includes nucleic acids, which is RNA and DNA, and
Right.
it includes
Right.
translation. So by that definition, for sure it would be under there. But then there was an exclusion that says anything that is used for an infectious disease, infectious disease pathogen, is not covered under the guidance. They didn't say it changes the definition; it's just not covered under the guidance, going back years. So and it's in the EMA
Wiseman.
documents, it's other documents as well.
There was at least promotional material out there that established that, for it to be a gene therapy, the mRNA would have to transform the underlying gene.
Well, so that was the spin. So they said that was the spin. So it doesn't integrate into the genome. Well, first of all, and I think I've got it here. Let me see. I think I have it. Here we go. So Peter Marks here, and this was in like a year ago in some kind of interview on an FDA channel or something. The person interviewing said, "I was wondering if you could comment on the thought the vaccines can particularly alter one's DNA." So they're not answering the question directly, is it gene therapy? They say, can it alter the DNA? Well, he says, well, no, the RNA can't get into the nucleus, and there's no mechanism for the RNA to turn back into DNA. Okay, that's called reverse transcript. Yeah. This is Peter Marks. This is the transcript over here. So I just pulled out, this is what I presented today. Well, actually, Peter Marks, and I told him respectfully, there's a paper here from the National Cancer Institute just recently that unlike his comment that says cells do not have the enzyme necessary to get RNA back into DNA. Here it is. The reverse transcribed SARS-CoV-2 can integrate into the genome. Okay. Now the same authors did another study where they made a distinction between virus infection, but not viral RNA transfected cells, which is sort of vaccine, if you will. But there's some dose issues with that. But the point is that the mechanism does exist, contrary to what Dr. Marks was saying. The second thing he said is that RNA can't get into the nucleus. Well, here's NIH with a paper, right, saying the title of the paper is Nuclear Translocation, which means something can get into the nucleus of spike mRNA. So not only the mRNA, the messenger RNA, but also the protein. Okay, and this is NIH. Okay, so here's two mechanisms. There are many others. Here's two mechanisms that said, okay, they can get into the nucleus. Now, doesn't mean they integrate into the genome. Doesn't mean they integrate into the DNA. But guess what? You don't need that to happen, because even if you've got DNA that's hanging around in the nucleus, even if it's not like inserted into the genes themselves, there's something called episomal transmission, which can occur. And these are two papers here. There's many others. So there are mechanisms that exist. Okay. And what you have to understand, if your listeners do nothing else and just turn off the interview right now, if there's one thing that they need to know, is that these products are more complicated than any other medical product probably that's ever been produced ever by anyone. Okay, there's so many moving parts here: DNA, RNA, lipid nanoparticles. There's so many parts of how genes are regulated that we don't understand. Just to make just to try and just brush it off in a very cursory way, it's just completely, this requires years of study. And we've discussed before, Pfizer's comment, or the person from Pfizer saying we flew the plane while we were building it. I mean,
we developed the plane while we were flying it.
Right, exactly. That's exactly right. So imagine you're at LAX, and the pilot says, "Welcome to the maiden flight of this new Boeing 999 airplane. Don't worry about the guys in the white coats on the wing with the duct tape and the Black and Deckers. They're just fixing a few things, but don't worry about it." Okay, imagine that. I mean, you would never.
She was in that paper, that article. She bragged about it.
Well, that's exactly right. They were bragging about it. So there's nothing to brag around. This is so complicated, and this is what we're seeing. And I just want to mention, you talk about efficacy. Sorry, I just want to go back to the DNA thing for a second and point out the presentation by Kevin McKernan, which I would absolutely recommend to anyone if you can get the link and put that as well. But to point out Kevin's unbelievable presentation, I mean, of course, I'm very familiar with his work, but he went right into the belly of the beast. He said, "Listen, guys, I'm a genomics expert. I've got patents on sequencing methods, and he knows his stuff. And look what we found. We found it by accident. We weren't really looking for it. We found plasmid, these DNA things, these DNA circles that are used in the production of the mRNA, but they should be destroyed or removed. But he's found chunks of them and big whole chunks of them. Okay, in the vaccines, and they've got stuff in there that shouldn't that we know would have been there, like an antibiotic resistant gene. But in the case of the Pfizer, there's something called an SV40 promoter. It doesn't matter what that is really, but it wasn't disclosed to the EMA. That in the right hand picture there, that's the circle. It's called the plasmid map, and that promoter wasn't disclosed. So this is DNA, and DNA. According to the FDA, I've got that here. This is an FDA paper here. Dr. Peden is a senior distinguished scientist at FDA. This was published in 2009 by FDA. And look at the highlighted section. DNA can be oncogenic. Okay, you don't have to have it integrated necessarily. Right. Okay, we don't. So there's so many things that we don't understand about this. And so if you're going to build a plane and you don't really know how all the parts work properly and how they interact with each other, that's what how does it, if you press this button, what happens?
So Dr. Wiseman, I have to interject here. So you and I know from different vantage points, but we both have been around FDA compliance nation, that for purposes of this emergency, the national emergency, the global emergency with WHO, the standard rules were thrown out. Although the propaganda tells us, that, we followed, good clinical practices, good manufacturing practices to the T. We know that not to be the case. Our hypothesis, best case scenario is that, you have this deadly pathogen circulating around the world. Governments are flipping out and they want to protect people. Okay, that's the best case scenario here. Now, but there are serious safety signals. Were those safety signals acknowledged at all in these meetings today?
No. I mean, the minimal. Dr. Offer asked the question. Dr. Offer Levy asked the question. He said, what's the safety issues? And so the only thing that came up was Dr. Mark said, I don't think there's any new safety signals. He threw it over to his Dr. Caslow, who's like the guy immediately under him. He said, can you just confirm that? He said, no, there's no new safety signals. And that was it. Goodbye. Next question.
So, that's problematic. At this point, a couple years in, over 2 years in, and we see significant data points around the world that we need to at least take a step back. That's extremely problematic.
Well, they won't even acknowledge that. They won't even say, okay, there's this analysis, but it's wrong for this reason. I mean, they've done it in a couple of isolated cases, they're talking about deaths and so on, but they haven't. But they really haven't acknowledged. Okay, there's this analysis in this paper. It's wrong for this reason.
Right.
At least do that. They haven't even done that. I mean, and again, I gave the example of the ischemic stroke that they were talking about extensively back a few months ago, and so proud that they found this thing in the VSD system. And they looked absolutely everywhere. They looked in every other database. They asked all the other regulatory bodies in the world that they knew. Can you see anything here? And they said no. And yet all we found, I don't know if I've got it in this deck or not, but we found it in the CDC's FOIA disclosure from January that there's a signal in that database by CDC's own calculation. What? It's your calculation, not mine. Why are you saying you couldn't find it anywhere? It's there. Okay. You can say, okay, it's wrong because of this and that reason, but don't tell me you couldn't find it anywhere and yet you did.
So here we are. They're going to recommend XBB 1.5. based on what you witnessed and heard unfold today, is it going to be business as usual? I mean, is this just going to continue, Dr. Weissman? We're not
Yeah, I
going to
mean,
step.
I'm afraid it is. I mean, you've got. I mean, I mean, the big. I mean, I think um, you've got CDC will present their data. Oh, yes, the vaccine works, and here's all the data, which is extremely limited for lots of reasons. And you have a study like this one. This is Cleveland Clinic. I'm sure you've reported on this one before. This goes back a few months, where they say this is not the only study to find a possible association with more prior vaccine doses and higher risk of COVID. That was a few months ago, Cleveland Clinic. Now, this week, just a few days ago, this is what they came out with. Listen to this. A few days ago, since the XBB lineages became dominant, adults not up to date by the CD definition have a lower risk of COVID than those up to date on COVID vaccination, bringing into question the value of this risk classification definition. So here we have Cleveland Clinic saying there's a bigger chance that you're going to get COVID if you're up to date than if you weren't up to date. Okay.
Yes.
Now, why are they not dealing with this? What's wrong with it? And
well,
so
I can share this with you, Dr. Weissman. First of all, that can't be cited as evidence. It's not peer reviewed. However,
it doesn't. Neither is.
I know.
Neither are Pfizer's studies, Moderna studies. They quote preprints all the time.
I knew that would get your blood flowing.
Moderna, by the way, Moderna put up their. I think it was pretty sure it was Moderna. Moderna put up some of their data. And they said, "Oh, it just came out in a preprint this week on medRxiv." Moderna put it up.
I understand.
Don't tell me. That's complete nonsense. I mean, that's complete nonsense. The whole it hasn't been peer reviewed. Okay, there's little that's been peer reviewed, and the stuff that has been peer reviewed. That's a very dodgy term.
It's a one-way standard, right? When there's data that doesn't help a particular narrative, they will say, well, this isn't peer reviewed. We can't be sure about it. But however, when there's data that bolsters or supports the narrative, and it's not.
We'll use it. We'll use Twitter. If it's a tweet, we'll use it.
Yeah, it's a double standard. It's just not right. It's very clear that there's a serious agenda that the regulators have been captured in some way, some manner. We suspect this. Okay. Us at Trial Site, you don't hear much about the origins discussion, do you, Dr. Weissman?
That's a whole nother.
I understand that. So my point is, there's a lot of investigation that needs to continue to happen. So, wrapping this up, I mean, you were able to present. You did a great job. You again, the breadth, the depth of what you presented, I was really impressed in just a few minutes what you were able to present. Any thoughts about for people that are studying this, that are concerned about this, that want to do something about this? What's your thoughts?
Well, I mean, you need to keep asking questions of your doctors, and ask your doctor, does he know? Does he or she know? She or he, whatever, know where the vaccine's going, where the mRNA is going, how long it's producing spike protein for, etc. But look at this. I want you to ask this question. We talked about this on the phone the other day, but this is from the FDA's briefing document. In the red highlight there, generation in the middle. Generation of immune escape variants may be further facilitated by chronic infections in immunocompromised hosts, or potentially by waning of immunity in immunocompetent hosts. Okay, all right. So that sounds like if they've been vaccinated, but if it doesn't get you here, look at the blue one. And as immunity increases globally from vaccinations and/or infections, natural selection of immune escape variants appears to be playing an increasing role in SARS-CoV-2 evolution. So what that seems to be saying to me is that when you've got vaccination, that is driving variants. So it's not like we're chasing variants. We're sort of almost if this
we're creating well, that's the Geert Vanden Bosch hypothesis that when you mass vaccinate into a pandemic, you're going to do exactly that. And
natural selection is
exactly. So they're now acknowledging that. That's a very important point.
I mean, are you reading that the same way? And look at that the blue.
Yeah, that's how And I saw that the other day when you referred me to it, that particular document. So yes, it would appear that many of the points, for example, in trial site news or what you've been reporting on or researching and talking about are coming true. And nobody listened to us. It was very difficult. We got censored if we were on social media. It's been horrible, and we're just simply trying to report on the truth. So I think you make very important points, Dr. Wiseman, that individuals, and that's what trial site's all about. It's about democratizing biomedical research. I mean, it's a very niche area, but it's a very big, important area. It's about democratizing that, translating it to a format and to content that people can understand, and doing it in such a way that is objective as possible. We want people to think for themselves. We don't want to tell people how to think. We want people to ask questions. So I think that's a very important point. People should ask their physicians. Physicians should start asking questions. I think that now, in terms of moving forward, there'll be a CDC meeting. Can you just quickly update us on what's next?
That's a really important point. So what FDA have done, they've kicked a certain decision down the road. Now, if you recall, sort of, the early days, 2 years, year and a half ago, especially when they were talking about the boosters, there were big discussions in the FDA meetings. Okay, which age group and which subpopulations and so on and so forth. And FDA have sort of got out of that business and said, no, that's the CDC are in that business. Our business is just to say, is it generally safe, effective, or whatever standard they're going to apply. But the CDC's job is to sort of define in more sort of granular terms which clinical populations should this be used in. So the discussion was there, but in a very abbreviated form, in terms of, well, just because these things may get authorized in a couple of months' time, does it mean that everyone needs to get them? And at
right.
least I think Offit was one of them, and there were others. I forget who, but they said, well, not everyone necessarily needs to be getting these in September because there's higher seroprevalence, there's natural immunity, etc., etc.
Yes.
But he said, but that's a discussion for ACIP, the CDC. So will they be having that discussion next week? I'm not sure. But at some point they'll be having that discussion. So we'll see. We don't have the full agenda yet, but that's CDC's mission. The other thing I just want to mention is that Novavax were there. Of course, Novavax, as you understand, is not a genetic.
It's not mRNA.
It's not mRNA. It's not a gene therapy product.
Yes.
And there were actually some of the public comments. There was a sort of quite a few of the speakers were Novavax advocates. It needs to be more available, and you need to authorize it for different ages, etc., etc. And one of the interesting things that I learned from Dr. Kaslow's presentation, that's the FDA's the head of the vaccine section. He said there was a meeting back in April of a committee. I forget the name exactly. A working group or something to do with developing protein-based, vaccines. And so I think they realized. I wasn't aware of that until today. I'm going to look at that meeting, look at the video of it. But I think in the background, FDA realizing, we're not going to be able to keep this up with these mRNA vaccines for too long. We've got to get back to some basics. So that was another point. The other point I want to point out that came up again, and I knew exactly who was going to bring it up because he's brought it up for the last several meetings, is Dr. Ofer Levy talking about, well, do we still have an immune correlated protection? We're measuring antibodies levels, and what does it exactly mean? And so FDA's Dr. Weir was sort of saying, well, there is a correlation between antibodies and protection, but it doesn't mean that's an immune correlate. It's different because we can't say okay, if you have this amount of antibodies, it means you've got this amount of protection. So that
Right.
so that again reared its ugly head, and should we be measuring T cells and memory cells and other kinds of parameters? And then the other thing that came up was okay, you're going to authorize these things in a couple of months' time. What should you be telling doctors to do right now? Should people continue to vaccinate the small number of people that are with the currently approved or authorized bivalent vaccines? And they said, well, they made a decision back in April that there was, even though the immunity had waned and so on and so forth, there was still some sufficient protection that was worthwhile, and so they didn't pull the vaccines. But at some point, they've got to pull them, and they've got to make that transition somehow or other. But I suspect they want to keep them on the books until the very last moment. They don't want to say, okay, they don't work, stop using them, because it's going to be harder then to reintroduce the next variant. They have to have
it's a problem, Dr. Wiseman, because we've put a story or two out about this. The current vaccines, I would argue, are not approved anymore because they were investigational. The monovalent was approved. The bivalent is investigational. Maybe you can argue the platform was validated one time, and therefore you can impute that approval onto the bivalent booster. But these are investigational products from our opinion, experimental. And so therefore, it's really problematic that these bivalent index variant plus BA4, BA5 is just hanging out there. And the standard message is, oh, it just works fine, and go get your shot, because it doesn't necessarily. Those strains are gone. They're extinct. And for all practical purposes.
Well,
so
they did say, someone asked a question. They did say, well, they do plan to transition in at least in the adult doses that they're going to roll out, to transition those to full-blown approval as opposed to the EUA. And the EUA, even though the pandemic thing doesn't exist anymore, the EUA provisions still do exist, and so they're able to still. But the thing you have to remember under the EUA is that the standard may be effective, and even though they may not have done any studies, okay, to show that it prevents this thing under this condition, all that is required is they have to have grounds for believing that it could be effective in these other circumstances. And so that came out in the response to the citizen petition that Linda Wasilla and Peter Doshi and Kim Witzak filed several months ago.
For the labeling, yes, very good.
And the labeling, and basically, FDA said, well, we don't really have to. All we just have to say is it may be effective, and we have to have some ground. So even if you show me a study that looks at a particular parameter, but you want it to really measure a different parameter, it doesn't matter. We just have to be able to infer somehow or other that the two things are connected, and you don't have to produce the data for transmission, for example. So it's sort of like a bit of a reveal. We sort of guessed that, but it's a bit of a reveal in the same way, similar way to the Brooke Jackson case, is that the judge said, well, it didn't really matter in the end, because even though it may have been fraud or may have been misrepresentation or whatever it was alleged to have been, it doesn't matter, because FDA can make whatever decision they want, even if the data are all wrong. So that's how they've been making their decisions.
Well, I would challenge that, though, Dr. Wiseman, in that the more that kind of inappropriate regulatory governmental behavior goes on, the less the public has trust in these very important agencies. And so you want to wonder why vaccine hesitancy is growing. You now know why.
Well, that's exactly right. I mean, they say they want to have trust, and they build trust. This doesn't build trust whatsoever. If they come out and say, well, actually, we can do whatever we like, and we've just been telling everyone, oh yes, the CDC have said on their websites, the FDA have determined these things, to apply their rigorous standards and the rigorous analysis, etc., and have determined these things to be safe and effective. No, they haven't. All they've determined is that they may be effective under the EUA standard, and may be effective it doesn't even need you to do a randomized double-blind clinical study.
Well, look, I want to summarize this for you. Again, you're doing great work. We're extremely proud of you. We consider you sort of a hero out there because, you do all of this on your own initiative. You're not working for some big company or agency that's paying you to do this. You're one of those people out there that have really made a difference. So I just want to recognize that and let everybody know that they should follow you. They should go to your Twitter, everything. You're basically declaring that the current vaccines need to be regulated as gene therapies. You heard it here.
That's absolutely. That's going to be. They want trust. That's going to be right at the top.
Absolutely, and I can't disagree with it because again, we've done the homework and we have seen that the companies themselves are describing them as that under their risk profiles.
Right.
Okay. Two, are these products misbranded or adulterated? That's a big question. Three, we need to address safety issues, equity in the vaccine injured. We partnered with React 19,
right.
Who's doing great work, and there are a lot of people hurting out there, and they need help, and this is inhumane to not help them, as well as with a lot of people struggling with long COVID. Full disclosure of safety data, sequence, and spike protein.
I think the sequence actually was disclosed, I think, last week or so in the back of a paper somewhere for Pfizer, actually. But anyway.
Okay, so we'll maybe do an article on that. No free passes to LNP.
Oh, that's a big one. Yeah.
This is a big deal. Lipid nanoparticles. We've read papers where already scientists are trying to look at ways to improve the mRNA vaccine so that they are safer. In regards to LNPs.
But by the way, whenever they can in the documents, they don't use the word lipid nanoparticles. They use the word lipid particles. They don't use the word nano, which might seem to be a very inconsequential omission, but it's a huge omission because FDA has a guidance somewhere about nanotechnology, and it's clearly saying, when you have the nano version of something, it's going to have different properties, and we have to. That's the whole point of making it nano, because there are different properties that get imparted by making these things very small. And
words matter.
Words matter. So I think that's a material omission of an omission of a material fact to accurately describe the product.
Absolutely. This next point is critical. I'll share with the audience. Long-term safety studies, what kind of cancer, autoimmunity, neurotoxicity. We're reporting on cancer cases now. They're very, very rare, but they're there. Okay, that's a problem.
Again, there's signals in the FOIA, in CDC's FOIA dump from January. There's according in the signal analysis, there's a number of 15 different types of cancer that signal, according to the calculation, there's a signal there.
Yes, absolutely. Restore trust in public institutions and science. 100% agree with you, Dr. Wiseman. And economic impact of vaccine adverse events. Very, very, very important. We don't know what that toll is. These are what we call externalities. These companies are passing their negative externalities onto the public, and it's completely inappropriate, illicit, maybe even a crime. Dr. Wiseman, we're going to wrap this up. It's always a pleasure. We learned so much from you. We'll invite you back when you're ready to update the trial site audience on CDC meetings, future FDA meetings, or other interesting or important news as it unfolds. So we want to thank you for taking the time.
Thank you. Nice to see you.